Genes are found on chromosomes at specific locations. The orientation of the genes with relation to one another and the distance between them can be determined. The distance is based on the frequency of crossing over between genes on the same chromosome. The closer the genes are to each other, the fewer the crossover events that will occur between them, and vice versa. 3-point test crosses are one way to determine the order of 3 genes on a chromosome, as well as the distance between them. A hypothetical plant has 3 linked genes, all of which control fruit phenotypes. A recessive allele of the first gene, P, determines purple fruit color, where yellow, P+, is the wild-type color. A recessive allele of the second gene, R, determines round fruit shape, where elongated R+ is the wild-type shape. A recessive allele of the third gene, J, gives a juicy fruit, where dry J+ is wild-type. To determine the order of these 3 genes on this chromosome and the distances between them, a test cross is performed between a triple heterozygote and a triple homozygous recessive. The offspring from this cross are sorted and counted. 2 of the phenotypic classes result from no crossing over between the parental chromosomes. These are parental phenotypes. The other 6 phenotypic classes result from either 1 or 2 crossover events between the genes on the parental chromosomes. These are recombinant phenotypes. First, the order of the 3 genes on the chromosome is established. One parent is homozygous recessive, so that parent can only contribute recessive alleles to the offspring. The other parent is heterozygous, with the 3 genes being coupled, that is, all wild-type alleles are on one chromosome and all recessive alleles are on the other. If no crossing over occurs, 2 types of gametes are produced, one with all wild-type alleles and one with all mutant alleles. These are parental gametes. Fusion of these gametes with the triply recessive gamete from parent 2 gives 2 parental classes of offspring, one triply heterozygous with a wild-type appearance, yellow, elongated, dry, and one triply homozygous recessive with an all-mutant appearance, purple, round, juicy. The order of the genes is determined by the progeny class resulting from a double crossover involving the 3 genes. A double crossover event changes the orientation of the gene in the center of the sequence. Since the frequency of a double crossover